after we have successfully divided our nuclear contents, now we have to divide the rest of the cell. So that stage is going to occur is called cytokinesis. Cytokinesis is going to be division of the cytoplasm. It does begin sort of overlapping with parts of mitosis. It's going to begin during telophase, but really you can sometimes see it going back as far as anaphase. This is going to be a ring of microfilaments contract at the midline of the cell, eventually pinching the cell in two. So here's an example of cytokinesis of an animal cell. What we're going to see is a contractile ring of actin microfilaments. They're going to draw around the center, and they're going to pinch tighter and tighter and tighter until eventually we have separated separated the glob into two. You can kind of think of it like, I don't know if you've ever used dental floss to separate a ball of dough. I mean, I don't know if you've ever played with bread dough or even Play-Doh, but you can start off with one ball, wrap a string around the center, and just pinch tighter and tighter and tighter until it separates, and the two balls become one. Or, I'm sorry, and the one ball becomes two. So this is the process of cytokinesis. Now, this is how cytokinesis occurs in animal cells, but it's going to be slightly different in plant cells. Brownie points for the first person who can send me a message or comment below about, well, no, don't comment below. Send me either a message via email or in our discussion groups about why and how plant cell cytokinesis is different from animal cells. Not just how, but I want to know why. Why can't animal cells separate their cytoplasm and divide into two cells in the same way that we do? And that's it for our discussion on cytokinesis. I'm going to back up just a moment so you can see where this is occurring. So here is a diagram of mitosis, well, the whole cell cycle. We have interphase over here where the cell is just kind of hanging out and doing its thing. During prophase, those chromosomes are going to condense down. That nuclear membrane is going to start to dissolve and those spindle fibers are going to form. At metaphase, we're going to line up right down the center of the cell. In anaphase, we're going to start to separate. So we're going to pull apart. In some cells, you can see a slight bowing or a slight pinching here in anaphase. This bowing or this pinching is what's known as a cleavage furrow. Cleavage furrow, that's going to be the indentation that is made by this ring of actin filaments. So we're going to pinch off and then separate fully in cytokinesis.